I just wanted, okay, I just wanted to spend a minute to thank everybody for, being here this morning and being part of the SFPM. As you all know, myself and Jesse Bowen from MIT were recently elected as co-president following, a very successful long tenure by Tony Letai. And we have an ambitious plan ahead, really trying to capitalize on the momentum that uh, functional precision medicine and models and data has right now, with the alignment of the NIH, the NCI, and the FDA. And so we're really looking at developing a new, strategic plan for functional precision medicine, expanding this community that you're all members of, and that job is not just us; it's on all of you as well. And obviously, we have some ambitious plans to try to put funders together and potentially come up with flagship projects that allow us to truly, act on the promise of functional precision medicine in the preclinical and clinical space. So this is work that's ahead, and as we build momentum around these, we have planned kind of a relaunch event that will happen during AACR. This will be our chance to kind of, discuss with everyone the future plan and where the society is headed, show, kind of our work on rebranding and solidifying our message and mission. And so please save the date. There is going to be an email that's going to go out later, but this reception will take place the Monday of the ACR. So that's Monday, April 20th from 5 to 7 p.m. The location that will be announced shortly. In addition to having me and Jesse there giving remarks, as well as members of, hopefully, all of the members of our board, you are also, there is also going to be Tony Letai. So, as you know, Tony was the SFPM president for, I want to say, probably close to 6 years. And he left this post when he became the NCI director. And so he's going to be there with us to give remarks during this reception. Obviously, there are limited spaces, and we will, give priority access to people that are members in good standing of the society. So if you haven't paid your 2026 dues or you are not yet a member in full standing, I think this is a great time to go and do that. It only takes a couple of minutes to become a member. So with that, I'll stop it here. Let me close my screen real quick. And I want to introduce today's speaker. It's really, our absolute honor to have Wes Tansley here with us today. It comes to us from Memorial Sloan Kettering, where he's an assistant member in computational oncology in the Department of Epidemiology and Biostatistics. He was a postdoc before at Columbia, and his lab is working on developing really cool probabilistic models and statistical methods, as well as machine learning algorithms for cancer. To look for new and effective combinatorial therapies to model tumor microenvironment and disease, and he also works on generative models for observational health records. His lab is supported by an Art 37 Merit Award, as well as from the Center for Tumor Immune Systems Biology at MSK and other awards. He's going to present to us some really cool data today, which I'm very, very excited to hear about his PAN preclinical project, which is an atlas of millions of drug response data points across models. And so without further ado, very much looking forward to your talk, Wes. Thank you so much for joining us.
Thank you so much for inviting me. It's a very kind introduction. Okay, let me go ahead and share my screen. Okay, everybody can see that? Good.
Perfect.
And let me
Yes.
see if it'll
Thank you.
still show up when I go to slideshow. Still shows up?
Yep.
Great. Okay, cool. So thank you so much, Alice, and Jesse, who's not here, and Aaron. So I'm going to talk about this work on what we call the PAN Preclinical Project. Just to give you an overview, I mean, Alice gave you a little overview. I'll just give you a 3-second overview of who I am. I'm a computer scientist by training, and now a computational biologist. And our lab works on machine learning methods to drive science forward. We have a lot of work on metabolomics, spatial profiling. I'm giving a talk at MD Anderson's AI and Spatial profiling series in a couple of weeks if people want to see some of our work there that's about to come out. Um, and we also at MSK have a huge annotated, digitized electronic health record that we have generative models over to learn patient trajectories over time. But today I'm going to talk about drug screens, which we've done a fair bit of work on as well for the last 5 years since I've been at MSK. And particularly, I'm going to talk about a core component of this platform that we've built that um, we're now translating into the clinic called AdaptX, where we merge some of our work on adaptive drug screens that we'll see how much time I have left. I might be able to talk a little bit about, and in combination with large external studies that we've gathered in this project I'm going to talk about, with some machine learning models that we've built to ideally create not just optimal drug data to train models, but also to personalize drug combinations. In particular, we're focused a lot these days on pediatric patients who have relapsed or recurred. And then down the road, potentially looking back on all that data to stratify patients and find biomarkers that could inform ongoing clinical or new clinical trials. So the ex vivo timeline, this is definitely a much more favorable group to this whole endeavor, I think, than I'm used to talking to, but and more knowledgeable maybe about this whole space. But the way I see this is, 2010, 2012, you had the Cancer Cell Line Encyclopedia, the Genomics of Drug Sensitivity in Cancer, which came from Sanger. The Broad and Sanger produced these large cell line studies, these atlases looking at drug screens across all of these like 1,000 or so cell lines. Very important work. I think tons of discoveries have come out of that. Certainly don't want to disparage cell lines. They're great models. Definitely don't take away from this talk that cell lines are not good models, but they do lack lots of important translational properties that would allow us to actually translate drug responses directly into the human host in terms of things like having to immortalize the cells, them being in 2D, and these monolayers, right? There's just so many different issues that come up. And so over the last decade or 15 years or so, people like Alice's group and others have developed better models in terms of patient-derived cell samples or organoids or PDXs. And I would say there's, at this point, a very sizable body of work that I didn't cite here because there would just be too many citations at this point of showing that there's actually a huge improvement in terms of the fidelity of predictions of human response if you use a more, let's say, ex vivo model than an immortalized cell line. And then, in clinical trials, looking at this in terms of can we use ex vivo models to inform therapy? Initially, people looked at this in blood cancers, where it's a lot easier to get access to these ex vivo samples. But then more recently, there have been a number of clinical trials, mostly in pediatric solid tumors, published in Nature Medicine in the last few years, that actually show that you can use functional precision medicine to inform patients' therapies for patients that have relapsed or recurred, and therefore have no standard of care in pediatrics. But as a machine learning person coming to this field, wanting to build models for functional precision oncology, there are no large-scale data sets that I'm aware of that really exist for drug responses to ex vivo models. And it turned out to be very difficult to try to find this data. And so that was an initial roadblock. But then also, when we go to the clinic, all patients usually get put on combination therapy because we don't want to create negative selective pressure and push the tumor towards a resistant state. We can hopefully try to find independent mechanisms of action that will lead to very rare rates of recurrence. And so we usually put patients on combination therapies, right? But almost all of these trials, in fact, all of the trials are about single agents. They do single agent drug screens. So if I have time, I'm going to talk about combination therapy discovery. But the primary focus of this is going to be on this first point of there's no large scale data set. So we can't really even start to build models unless we're going to try and just take cell lines and somehow adapt them. So there's no large data set. So we built one. And so what does this look like? We built this project that we call the Pan Preclinical Project. It's a large ex vivo drug screen database. So this is drug screens on organoids, patient-derived cells that are in short-term culture. There are PDXs or PDX-derived cells. Basically, anything that's not an immortalized 2D cell line is what we took. We found 26 published studies. And more or less, my job in this, first of all, this is all led by all of my trainees in my lab, and they've done phenomenal work. And my job has been basically just to politely harass all of the different people who did all of this real work in the 26 published studies to ask them for their data. It turned out that more than two-thirds of the data, two-thirds of these studies actually didn't publish the original data because the standards in this field are not like genomics, where the editors say, okay, submit your published drug screen data here in this format. Like we don't have those standards in this field, and so a lot of this project was about how do we standardize this data and make sure that we have enough raw data to actually harmonize across all these studies. So what we've built with PPC is this large pan-cancer data set. We sometimes had omics, although I would say not as much as we wish we could have. Some of which is just because a lot of these studies focus more on the drug screens than on the omics, and some of it is because of regulatory issues and privacy issues, like GDPR prevented us from getting a lot of genomics information, for instance. So we've got 3,000 samples in this data set, not all of which have drug screens, but 3,000 samples overall. Either they have drug screens or they have omics on ex vivo models. It's a pan-cancer data set. It includes both adult and pediatric cases. And there are 3,000 unique drugs as well. And in total, over 2 million unique viability measurements. So this is all focused on viability measurements. Again, we took the viability measurements however they were done. So some of them use cell titer glow, some of them use microscopy. There's lots of different ways one could measure viability, which, as you might imagine, introduces some batch effects. And then I had a medical student who went back and worked with a lot of these authors to develop harmonized annotations for both the primary site and if the sample was taken from a metastasis. She actually mapped out where that metastasis was taken from and where the original primary site was so that we could actually get at the difference between primary mets. And I'm going to talk very briefly maybe about that, but I'll leave the rest of the paper. So what did we do with this data once we had it? Like I said, we're a machine learning lab, so we did machine learning things. That's what we know how to do really well. So we built a machine learning model to try to understand, can we actually take this large heterogeneous collection of data? So, some studies said, I want to measure 1,000 drugs all at one concentration. Some studies said, I want to measure 30 drugs at 7 concentrations, right? So we have to harmonize across all of that. And so what we did is we built this giant non-parametric Bayesian tensor factorization model that includes some contrastive learning. So effectively what it does is every sample gets an embedding. Every drug gets an embedding for every single point along a very fine-grained dose-response curve. And then we inject this contrastive learning penalty to kind of incorporate our prior biological knowledge. So for instance, if 2 samples both came from the brain, we would assume that they would have some sort of similar sample embedding. And similarly, if 2 drugs have the same mechanism of action or drug target, then we would try to force those embeddings to be a little closer. But these are very sort of flexible things. These aren't hard-coded. They allow the data to kind of overwhelm. And so if the data says that like actually these drugs are very different, then we let the model learn that. Okay, so once we've done that, can you actually predict across all these different cancer types on held out data? It turns out yes, you can predict very well across different cancer types. You can predict very well across different drug classes. And I would say that and we have data showing this, so I don't have it here, but that basically the sample size is what really is driving the correlation here. So the model predicts well across a huge number of different cancer types, but for instance, we don't have very many kind of generically labeled sarcomas that don't have a very fine-grained label like Ewing sarcoma or osteosarcoma. So yeah, so we have a model. It can predict well. And it turns out the first thing you might ask is, does every data set that we add here actually help? So these are the 26 studies that we incorporated. And you might ask, like, is a pancreas cancer organoid data set from Dave Tuason really helpful to, a glioma data set from a group at Samsung, right? It turns out that they are, right? So every single data set, so every dark blue star here with dark blue square with 3 stars in it is a highly significant. If I hold that data set out, and then I look at how well does the same model perform without that data set incorporated, if it's dark blue here, it means that that data set is less accurately predicted without that data set than with it, right? So effectively, like, the ones on the row here are helping the ones in the columns. And so basically every single row here has a bunch of stars in them, meaning that they help lots of other data sets to incorporate them. Kind of counterintuitive, but it turns out to be true. And in fact, it tends to be that actually the ones that are more sort of focused studies on specific cancer types get a lot more help from incorporating other ones than the ones that are sort of like very broad. So for instance, like this lead one here has a lot of different cancer types in it. We actually can show that you can learn interesting rational embeddings. So if we look at a UMAP of the drug embeddings, for instance, you can see things like MEK and ERK cluster together, PI3K and mTOR cluster together, HER2 and EGFR cluster together. And if you really drill down into it and look here and pick your drug of interest, I think you'll generally see that there's a lot of local similarity between very rationally coded, chemically coded drugs here. That's interesting. The model has learned some interesting chemistry and biology. And it turns out that actually the sample embeddings also cluster rationally, both in terms of the actual cancer indication, so whether it's pancreas cancer or colorectal cancer. But even beyond that, things like breast cancer and ovarian and uterine cancer, they actually cluster somewhat close together, which is a function of the fact that they both respond similarly to estrogen therapies in culture. So there's some interesting kind of cross cancer type clustering as well. Do these drug screens actually tell you something about the underlying biology of the ex vivo samples? It turns out the answer there is also yes. So, for instance, we can look at the samples in that were treated with Nutlin. This is from Beat AML, and we can ask, so for these samples, if they had a p53 mutant, do they actually respond worse to Nutlin? And this is all predicted data, right? So this is held out predicted data. So. And it turns out, yes, they do. And then if you can even break it down even further, and you can show that for some of the things like FLT3, that actually what the model has teased out here is that there's really no difference between responses in the mutants when there's a discontinued drug that targeted that mutation, whereas for the ones that have been approved in the clinic, they actually do see a significant difference here. So we're learning some interesting rational biology, and this is without using any of the omics. So we held out the omics and all of this as sort of an orthogonal validation. So then can we actually understand things about biology here? Again, yes, I think the answer is yes. So generally what we see when we look at these sort of like mets versus primaries, and I'll again say that, so one complication here is it's very difficult to get a paired same patient met with the original primary. For lots of reasons, right? Like most cancers, or a lot of cancers, patients come in already metastatic. We don't know what we don't have access to their original primary before it metastasized. There's a lot of issues there. And so what we have is mostly not paired same patient, but rather population level information. So we have a lot of primary samples from a lung adenocarcinoma, and we have a lot of brain mets that originated from lung adenocarcinomas. And so what we're asking here are questions about, at the population level, do lung brain mets, how do they look compared to original primary lung cancers? And so, unsurprisingly, probably, maybe as a positive control, in fact, yes, metastases are much more resistant to drugs. Often the patients have been pre-treated by a large number of lines of therapy by the time they're metastatic. And so we just see a general shift towards a more resistant state. Then you can also ask that, if I drill down into standard of care drugs, are they going to be more resistant to standard of care drugs in the MET case? Yes, again, they've mostly received standard of care drugs by that point, so it makes total sense. But what's interesting is that if you Z-score these things and you take away that kind of baseline resistance, you effectively could think of it like subtracting away the mean AUC for a given sample. On a relative basis, so in terms of how do these samples change in terms of rank, right? How do these drug targets change in terms of rank of whether this drug target is going to be effective in a lung adenocarcinoma primary versus a brain met from a lung adenocarcinoma? It turns out that just looking at mets versus primaries, you actually do see a large number of drug classes here that actually tend to be, on a relative basis, more effective in the, or more toxic, I should say, in the metastatic case than in the primary case. And similarly, there are also some drugs that go the other way. So then we can ask questions about cell lines. So what we did was we went back and we incorporated all of the large cell line databases. So GDSC, CCLE, all of DepMap effectively, PRISM. We incorporated all of these different cell lines, which is something on the order of what we had in the PPC project originally. So it's like another 2 million or so samples or observations. And we can first ask questions about, okay, so if we run this model, again, with these contrastive penalties that try to make it so that samples that have the same primary site, for instance, should be clustered similarly, it actually we actually find, and this is the same UMAP space, we're just splitting it out left and right for comparison. You see that the ex vivo samples still continue to more or less cluster by cancer type. It's a little confounded because we've added in all these cell lines, but we basically just don't see any of that structure popping out here in the cell lines. The cell lines tend to be more or less driven by their underlying molecular alterations, and have really potentially lost that origin phenotype that they have from their original tissue site, which maybe for cell line people is maybe not super surprising, but I think is important when we think about how will drugs work in these patients where they are having these cells in their actual original tissue sites. Um, then what we interestingly see is that there are actually systematic biases and differences in the drug responses. So cell lines are actually more sensitive to a whole class of different drugs systematically across cancer types than their corresponding ex vivo models. So the way to read this is, if it's red, it means that a cell line is more sensitive than an ex vivo model. When we look at, say, AML, this is how many AML samples we have there that are ex vivo samples. We have a lot because Beat AML was a huge study versus how many cell lines do we have that were originally AML annotated in Cellsaurus. And so all of the red here means that this drug class, if I look at the average Z-score for that drug class, so again, taking out whatever baseline resistance or sensitivity things might have, that this drug class is actually more effective in cell lines than in ex vivo. In blue would mean the opposite, that ex vivo samples tend to be more sensitive. And so what we see is that just generally speaking, maybe as a biological positive control even, you would expect that these cell lines, which have been selected to be cells that are highly cycling, so that they can grow quickly in a lab, they actually tend to be targeted much better by these drugs that are targeting cell cycling or various points in cell cycle or DNA replication, these kinds of things. So that's one interesting thing. And interestingly also, we also see a systematic shift in the other direction towards things that we're kind of calling cell signaling drugs. So things like, for instance, TGF beta, TNF alpha, HER2, right? These are signaling pathways that are signaling targets that potentially, not necessarily saying always, but potentially could be shed by a cell line, or at least have their downstream impacts impacted by a cell line. And so I think this has really important implications for when people try to go and look at a cell line for translational drug response, as opposed to looking at an ex vivo model. This is telling us that really there are some fundamental differences in how these things respond to drugs, and that should really inform how we think about functional precision oncology if we're trying to just use cell lines as our big data set. Now, when I gave this talk to one of the breakthrough cancer disease teams, Jesse was in the audience, and his question was like, well, isn't this all just explained by media? How do I know this isn't all just differences in media? So it's true. Typically, ex vivo models use very different additives in their media. And often many of them are the same ligand as the drug is targeting the receptor of, right? So like EGF, and you get EGFR inhibitors. So maybe everything is just explained by the differences in this. And so Jess, who's the co-first author on this paper, did the valiant work over the last couple of weeks of going back and actually pulling out the 50 different additives that were used across all of these 26 studies, plus all of the different cell line data sets. So this is all of them that we could find. Maybe I'll take a pause for a second in case anyone's like, wait, are you considering the additive that I think is really important? We consider a lot of them, IGF-1, PDGF, a lot of the things that you would think as signaling things. Some of them are sometimes kind of weird, right? So like Nutlin-3A was used in one of the ex vivo studies, for instance, as a thing to give to, for some reason, the mutant p53 samples. I'm not a wet lab person. I've never touched a pipette. I don't know why they need to give this drug for this thing. I don't know why any of these additives are necessarily used. But this is sort of across all these 26 studies, these appeared at least once. And then I think 42 of them appeared in at least 3 studies, which is sort of our cutoff for like whether or not we're going to look at it. I will say also this is a highly collinear set of additives, right? So if I were to just look at building one big linear regression model here, it's collinear. So like, you can't really fit it. The covariance matrix would be degenerate. So, but what we can do first off is we can ask the question of, so if I build a simple two covariate model and I say, okay, consider what is the z-score of this model for this sample when I know whether or not it has this additive and whether or not it's an ex vivo sample. Right, those are my two covariates. And then I look at whether that additive changes the coefficient on the ex vivo sample. Right, and so effectively what you're seeing here is that this is the coefficient on whether it's actually whether it's a cell line or not. Right, so what you see is that bigger ones, these bigger coefficients here correspond to the cell line actually is impacting the underlying z-score. And so this is effective, and this is ordered in the same order that I had before, showing that effectively the pattern remains effectively the same. So any individual media additive is not actually accounting for these actual drug responses that we see systematically. So it's not that. Similarly, we can look at the bottom here and look at the ones that the ex vivo samples are more sensitive, and we still see that the additives don't seem to correct for this. Now you can also build a very big regularized linear model to control for everything simultaneously. Very difficult to actually get anything really significant out of that because there's so many different collinear variables. But we did that. So this is a lasso model with like 100 bootstrap resampling. And we still see the same trend. So over here are the drugs that originally we said were cell lines are more sensitive. Over here is the ones where we said the ex vivo are more sensitive. And this is what controlling for all of these additives simultaneously would say is the effect of being an ex vivo versus cell line sample. And we still see the same trend. But again, very difficult, collinear. Like I'm a statistician, like I wouldn't really put a lot of faith in the ability to do this outside of like some very, very strong signals. So I think it's so reassuring that we see the overall signal, even when we control for things in this underpowered data set to do that. Okay, but again, I don't want you to walk away from this thinking that cell lines are bad. Cell lines are great. And in fact, it turns out that even though they have these biased drug responses, you actually still get predictive performance improvement if you go and take out those cell lines and incorporate them back into the overall data set and refit the model. So everything that you see in green here shifted to the right of this line is a sample that's predicted better on that drug class if you use the cell line data as well versus if you just use the ex vivo data, right? So basically just better data is always better. That's the lesson from machine learning of the last 20 years. Just give me all of the data. Don't worry so much about like how biased it is. The machine learning models are very good at sorting these things out. And just a lot of my like rich dependency structure still remains in the cell lines that allow us to understand better how to predict samples that we see some drug responses on when I've incorporated all this cell line information. So cell lines are great. We love cell lines. Okay. And then we've done now kind of going forward. So that was a factor model. It's not super great at like taking a new sample because it's a factor model, sort of assumes a fixed data set. So kind of thinking ahead now, what we want to do is build this sort of foundation data set that we've got, build on this foundation data set to build a foundation model that can actually be used to guide new studies. And so we've done that. So this is a large transformer model, kind of inspired by BERT, as many transformer models are, which really focuses on this idea of few shots. So I would say most of the predictive models in drug screens are kind of assuming what you have is, oh, I've done bulk RNA-seq, I've done whole genome sequencing or some sort of sequencing. I'm going to feed that into a model and make some sort of prediction about how drugs will respond. I think for a lot of functional precision oncology cases, for a lot of reasons, like genomic sequencing takes a long time. Sometimes that kind of like closes the therapeutic window, for instance. It's also much more expensive to have to do all those omics. It's difficult to know how to get RNA-seq that's harmonized as you go through this thing in the clinic. So there's a lot of problems, I think, with doing that if we want to translate this into the clinic. So this is why we've built this model as this few-shot model that can specifically just take in drug responses, right? So a thing that you can use for a patient where they undergo resection, you screen their drugs, you screen their samples across a panel of drugs, and then within 7 days, you can just take those results, pipe that into this model, and for instance, maybe you had very few cells from this patient, and so you didn't get a full range of drugs that you want. So this model is able to, for instance, impute a lot of those missing drug responses. You can use very few doses. You don't have to do a huge dose range. You can impute those very well. And the idea is you can ask it, so how would a new drug respond, or how would a different dose respond, create a response? Inner workings, again, this is just a big transformer. that's structured in a clever way that my postdoc Antoine did. And then it gives you this embedding for every single patient as a function of just seeing the drugs that their sample has been screened on. And from that, we can predict the viability of some new query drug or dose. And we can show that, in fact, if we look across different machine learning baselines, like you might use XGBoost to do this, or maybe something a little bit more sexy, like a foundation model that was published in Nature, like TabPFN, we can show that this model actually outperforms all of those. And what this really means is that as you go up a number of experiments, in order to get the same amount of predictive power, so for instance, over here, 50 with this fine-tuned version, which we call FM++, versus 100 for kind of all the other methods, that means we can predict as well with half of the samples, right? So half of the tissue, effectively, we can save you. Okay, so that's great. We have a predictive model. But it's also a foundation model, right? So the benefit of the foundation model is not just that it's robust and can predict on new held out data sets, but also that the embeddings can potentially tell you something more than just the original task. And it turns out that that's also true for our foundation model. So we can actually use the foundation model embeddings to predict genomics and expression. So this is, again, BEAT-AML, because BEAT-AML is just a very large cohort, and they did lots of omics for all of their drug screens, which is why we kind of use it a lot. And so what we can do is we can take that BEAT-AML, and this is a model that was not trained on BEAT-AML, right? So this is all out of study predictions. And what we can show is that, in fact, yeah, I mean, for the mutations, especially that matter in in AML, like KRAS, like NPM1, we actually have a pretty good AURC on predicting those mutations. And then similarly, we can look at lots of different hallmark pathways and aggregate them using principal components and look at sort of the first principal component of these gene pathways and show that we can highly accurately predict a lot of, again, the important ones like EMT that you might expect, and like KRAS signaling in the BEAT-AML cohort. So it's pretty cool. Like, so the idea is that you can effectively use this to provide some sort of biological rationale that might make a molecular tumor board more comfortable with recommending a treatment based on a drug screen result. And you might also ask, like, okay, so you can do this at a bulk level, but can you actually do it kind of in tandem, right? Like, do the genomics make sense in the context of the transcriptomics that are being predicted and in the context of the drug screen that's being predicted? And the answer there is yes. So what we see is that, for instance, KRAS mutants, if we look at the true KRAS mutants in this data set, there's 18 of them out of versus 404 wild type. They're predicted to have a much higher probability. Does it also predict that KRAS signaling will be up in those mutants? Yes, it does. And does it predict that, in fact, something like trametinib, which is targeting MEK, is going to actually end up with a more sensitive response than the wild type cases? And the answer there is also yes. So there's a lot of biological rationale there. And there's a similar story here for NPM1, looking at apoptosis and sensitivity to venetoclax. And again, just kind of like provides a nice biological rationale that we can kind of like automatically predict for all these patients as they come in. And now we have a website, and actually this is live as of today. So if people want, you're more than welcome to. Let's see if I click this. Yeah, so you can actually go here, and it's live. You can explore this data if you're interested. We have lots of different things you can do with it. You can look at the UMAP. You can compare drug responses. So this is, so like you can look at drug responses in terms of the comparison to cell lines. You can look at curves across different drug classes. So it's panpreclinical.org. Encourage everyone to go there and play around with it. Would be very interested in understanding what it is you guys would like to use this data for, or this portal for. We can very quickly add new features to it. So if people are interested, definitely would encourage you to go take a look at the data and take a look at the portal and let us know what you think. So that's sort of like the high level overview. This is more of like a I'm turning the data over to you guys kind of thing. So why don't I maybe move on from this and just briefly tell you. So this sort of solves these two problems, and I'll just take a few minutes to maybe briefly go over how we think about doing these drug screens in this platform that we're now trying to translate into the clinic, which is this problem of combination therapy discovery. And this all comes from a paper that we have in Nature Communications last year. So we have these studies which includes our database, that are almost always single agent. And almost always we put patients on combination therapy. So this is a trial, a phase 3 trial that succeeded on adding an additional drug to NCHOP or to RCHOP for diffuse B-cell lymphoma. So, up to 6 drugs at a time can be highly rational. But, when we think about finding those combinations, it's very difficult. As you scale up the drug library, you think about the number of pairs or maybe even triplets that you might want to find. There quickly become more experiments that you have to do than there are atoms in the universe. And so the question is like, how do I find those? And there's maybe 2 classical approaches. One is be a very smart biologist who really knows what they're looking for, have a small library of drugs that you think are important, look at all those combinations. Or you can take a large library and just do a really unbiased screen of a bunch of random combinations, usually almost always done in pairs. I've never seen anything large scale that's not pairs. That will change soon. But our idea was really to use active learning to design very quick, small adaptive experiments that really guide things much more efficiently. And so the idea is you start with a large library, but you do small experiments. But then when you get that data back, you feed that into a model and you use that model to design a much more informative next set of experiments. And we can actually prove that we can optimally explore the information landscape here to train effectively an epsilon optimal model, meaning like within some small epsilon of whatever some sort of oracle could have designed. And then the hope is that that really optimal model helps you discover better and higher efficacy drugs. So we have the system. It's called BACHI for Bayesian active therapy combination hunting via iterative experimentation. And the idea is really the standard active learning setup. So you start with some sort of seed batch, which I'll talk about briefly for a second. In a second, you do that initial experiment, then you enter this active learning loop. Sometimes people call it a lab in the loop approach, where you build a Bayesian model. It can be any Bayesian model, actually any model that gives you valid uncertainty quantification. It doesn't have to be Bayesian. And then what you can do is you can simulate from that model. And you can say, okay, if I were to try this sample with these drugs and I got back some result, how would that change my model, right? And so effectively, if I do an experiment and my model doesn't change at all, it's not a very informative experiment, right? What you want is to choose experiments that whatever the result is, the model will change drastically in one way or another. And that might be informed not just on that drug, but on lots of other drugs, right? Like response to this drug might inform lots of other drug responses. And so you can quantify that sort of uncertainty reduction in something formally called information gain. And then you can measure that information gain across all the different possible candidates and drugs and samples you might try next. And then you can use that to design an informative batch. And we did all of this as a proof of concept using cell lines, but now we're adapting this in the clinic to do it with ex vivo samples. And then you run that over and over again in this active learning loop until usually you run out of money or you run out of time or samples. And then you do a validation batch where you say, okay, I've got this optimally trained model and I use it to predict some hits. And I'm going to validate them, and then I'm going to maybe put them in mice and validate that those actually still work. How do you solve this cold start problem? So you need some sort of seed batch. The question is, how do I optimally choose this batch when I don't know anything about this sample that's coming in? I won't go into it too much. Effectively, what we do is we do this k-meteoids thing, where rather than just sort of like random selection, which would end up with like kind of clumpy things and kind of like miss a lot of these sort of outlier areas over here, you can do k-meteoids, which kind of nicely covers the space through this optimization approach. It turns out that this works much better. So k-meteoids is over here in terms of mean squared error, as opposed to choosing random drugs is over here. We choose dosing based on sort of like looking at the second derivative of the dose response curves that we see. Won't go into that, but we have a nice little heuristic that I think works quite well. And how does this Bayesian optimization or Bayesian experimental design work? There's 20 pages of learning theory that Christosh in my lab wrote for this paper, so I'm not going to go into that. I don't understand half of it, but Chris is brilliant, and I'll just give a high-level overview of how it works, which I think is kind of intuitive at least. So the idea is you have a posterior distribution over all the different possible true parameters of the model that you're using to predict these drug responses. And what I can do is I can say, okay, what I want is to choose the samples for which if I choose any point in this posterior distribution and I draw a line between them, I want that line is going to shrink the most after I run this experiment, right? That I want this posterior distribution to contract optimally, right? So it's like this weird space, and I want that space to like maximally contract. And it turns out that the max is actually that doing the max is what you want, but doing the average contraction of like pick two random points and look at how close they are. It turns out that that's actually just as good within some small epsilon again. How does this work? So if we look at the large cell line combination studies retrospectively, works really well. So we can ask, for instance, Almanac, GDSC squared, Merck. These are all huge landscape studies, but also still sparse, right? Because you can't explore this space exhaustively. It's just too expensive. And so we went back and we said, what if we were to design these experiments using Batchy as opposed to say like a random ordering of these plates? It turns out that if I run 15 iterations of Batchy and I run 15 iterations of randomly designed experiments, and I look at how well the models predict the held out data, our models predict much better when we do Batchy optimization as a Batchy design instead of random designs. This is also true if you use other basic active learning baselines like variance sampling or information gain or expected information gain. But I think what's really cool for, especially for people looking to do these experiments, is number one, we can show that we can actually save hundreds of thousands of experiments in some cases. So if you look at GDSC squared, if I look after 15 rounds, and the question here is the y-axis is how many more randomly designed plates would I have, or randomly designed experiments would I have to do in order to get a model as good as the Batchy model? And the answer here is like 125,000 experiments to get additional experiments to get as good of a model here. So we can save you all of that time by just really intelligently designing your experiments. And I also think what's interesting is that it kind of encourages something that's maybe counterintuitive to biologists, which is actually the more ambitious you are, the more you gain here. So rather than saying, let's start with like 5 cell lines, it actually, you get a much bigger gain in terms of how much you can save in your experiments as you add more and more samples, right? This is equivalent to like, I can recommend movies much better if I have 1 million users as Netflix, as opposed to like just asking 20 people that randomly. And so, yeah, what we see across all of these is that if we take the number of cell lines that we have and we start to scale them down, the efficiency gain really goes down. So the more cell lines, the more drugs that you have, the better that this system is going to work. So we validated this prospectively in cell lines, as I said. I won't go into too much of the results here. Maybe I'll go through it a little quickly just for interest of time. But this is effectively would have been, if we had done this exhaustively, the largest combination drug screen ever in terms of like the library size of the drugs. It's still the largest in terms of library size, but we just didn't do all the experiments. Instead, we ran 15 rounds of Batchy, 54,000 experiments. So a very, very sparse, 96% sparse observation landscape, focused mostly on pediatric sarcomas, which is where a lot of our collaborations happen right now. And what we see is effectively a nearly optimal predictive model, so a Spearman rho of 0.91. The noise that you see around here is really just standard high throughput screening noise that you really can't reduce to zero. And even though our Batchy model is not designed specifically to predict synergy, and synergy is extremely rare, as everyone knows. So we looked at over 3,000 randomly chosen combinations, and we only saw about 13 that were confidently called as these are definitely synergistic. Batchy has a very high AUC at predicting those out of sample. We did this prospectively. I'm going to go through this very quickly, but the idea is that we don't want to just select toxic drugs, right? We want to select drugs that have some notion of a high therapeutic index. So we use control cell lines that try to mimic various human tissues, and we look at whatever our target cell line set is, and we ask, like, what's the gap here in the viability? We want that gap to be really high. Across our 5 Ewing sarcoma lines, what we saw is that our predicted hits for Ewing sarcoma in terms of therapeutic index, as compared to all of the 54,000 experiments that we did otherwise. So the median of those predicted hits were about as high as the 99th percentile of all of the other data. So very, very highly efficacious drugs. And in fact, 3 of the 6 phase 2 trials at the time for combination Ewing sarcoma therapies were mechanistically the same as our top 10 hits. I'm going to maybe skip over this. These are the drugs we chose. You can see the paper if people are interested. There's also some interesting results on osteosarcoma. For those who work on sarcomas, maybe it's not very surprising that osteosarcoma is very difficult to find combination therapies that work across all osteosarcoma cell lines. Extremely different, right? From erythrosis, they're all kind of crazily rearranged. there's maybe like 20 different molecular subtypes of osteosarcoma, as opposed to Ewing, which is much more homogeneous as a disease. But we were able to at least show that we can actually do personalized predictions in terms of highly resistant, resistant, sensitive, highly sensitive. And then maybe one brief comment that I'll say is we also asked the question of, are pairs enough? So in our Ewing experiments, we saw that talazoparib and topotecan was the highest predicted outcome, then topotecan and mitomycin, and we also had talazoparib plus mitomycin. Right? So these are three different drugs. Basically, all pairwise combinations of them were in our top hits. So the natural question was, what if we combine all three of these? So we did that experiment and asked, like, is it enough to just like predict it on pairs and see if those pairs then generalize to be good triplets? So we looked at a huge grid in 3D, and then we asked in the simplex space of like, how much drug should I put into each of these different agents in order to get the maximum therapeutic index, where hopefully you would see something in the center here or somewhere in the interior here, which would say that all three drugs are helping. And so are pairs enough? The answer turns out to be no. That actually, when you do all three of these and you look for the best thing in terms of this therapeutic index measure, mitomycin basically gets set to 0. It's 1 nanomolar. So more or less nothing here for mitomycin. But sometimes you do get lucky, it turns out. So we were actually able, for two of the cell lines, for osteo cell lines, to actually show that talazoparib, trametinib, and alisertib, so these like aurora kinase and MAC and PARP inhibitor combinations, for two of these 4 cell lines that we ran on osteo actually do seem to have non-trivial combination efficacy. And that's coming just from looking at pairwise combinations, right? So you can get lucky. But we kind of think at this point that if you want to find higher order combinations, you have to actually screen higher order combinations. And so we're doing that now with desmoplastic small round cell tumor, which is a very large fibrotic abdominal tumor, almost always occurring in young adult or adolescent males for reasons we don't know, disproportionately African descent for reasons we don't know, but basically just a dismal standard, a dismal set of outcomes, 11% 5-year survival. And it just so happened that when we started working on this project, a patient, Maurice Campbell, unfortunately died of this disease, but was through a generous donation, we've created a Maurice Campbell initiative to work on DSRCT and to fund some of this work on what would otherwise be a very difficult to fund ultra rare cancer. And how does this work? So for us going forward, we've added some DSRCT lines that we have internally and we've done some more rounds of batchy as sort of a bootstrapping for ex vivo samples. We've actually got a workflow that takes tissue directly from the biobank, bypasses biobank rather, directly from the OR, straight to the lab here in Andrew Kung's lab. We've done some initial pilot studies designing pairwise plates that maximally discriminate between various types of subtypes that we see. And we're doing triplet screens as well to try to do this idea of maximizing therapeutic index. We've done pilot results already in 2 patients, 3 samples. And what we see in these pilot ex vivo samples is actually we do see that there are triplets for which we find very good therapeutic indices. So this is 0.4. How good is 0.4? This is kind of like an arbitrary number to most people that haven't read this paper already, but effectively it's about as good as the 95th percentile in Ewing sarcoma drugs that we found. And it's about as good as any of the osteosarcoma hits that we tried. And then we actually also see that some of these triplets actually improve quite substantially over the underlying like best possible 2 out of 3 drugs that we could have chosen from that triplet. So there really does seem to be some valid gains here from doing triplet drug screens instead of doing just pairwise drug screens. And so we're thinking of this like 0.4 therapeutic index as sort of like a go, no go kind of hit selection for clinicians. I'll just say we've got over 40 patients that we've already banked live viable cells on this. We're going to accrue 30 more. And then this year, we're hoping to launch a pediatric precision medicine trial for all relapsed and refractory pediatric solid tumors at MSK. Okay, so I will just wrap up and say thank you so much. This paper has, I think, 40 people that we acknowledge as helping us gather the pan preclinical data set. So thank you so much to all of you. I know I bugged you a lot. It's very much appreciated. We even had some companies that helped us, that gave us some of their data. And so thank you so much to all of those people. And then thank you to everyone in my lab. So Antoine was a postdoc who built a lot of the foundation model. Chris built the original factor model. Annalise is the med student who helped us. And then Jeff, Jess, and Carl are the three first co-authors of the paper, which will be on bioRxiv in, let's say, I don't know, a week and a half or so. And so that's all. Thank you so much.
Thank you. Thank you so much, Wes. This was great. And I know we have a few questions. We have a little bit of time for questions. We have a few in the chat. So if anyone who put a question in the chat would like to speak up, that would be great. I will just add that I registered for the website as you were speaking, and it's actually really fantastic and intuitive. Lots of data. Absolutely. I will be exploring that. Sandy, do you want to go?
Sure. Very nice work. I really enjoyed it. I had a question. When you showed the clustering of drugs, you showed a very nice clustering based on mechanism of action. I was wondering, do you see that on all tumors? The reason I'm asking is that when we've done a screen on primary tumors, and there's a sort of very narrow focus, is just in fibromyalgia carcinoma, there was no clustering based on mechanism of action, but there was a clustering based on pathway by which the agent was metabolized. So, for example, everything was metabolized by CYP3A4 worked well because CYP3A4 is down about 1,000-fold. There are a few other metabolic pathways like that. So, have you seen anything like that, or do they always cluster by mechanism of action?
Yeah, that's a very, very wise question. Like someone who's done this a lot. Yeah, we also struggle with this, and we struggled a lot with this in the Batchy paper as well, where I think intuitively everyone is just like, you just know the mechanism of action of the drug. You can use that to target the alteration or whatever the actual target is of the drug. And we just never saw that, right? So importantly, these drugs do cluster by target. I wouldn't call them necessarily mechanism. Like they target, like by their intended target, they do cluster. Every drug can have multiple targets, obviously. So they do tend to cluster. To some extent, we're enforcing a little bit of that clustering. I think the local structure is the most interesting part of it. But definitely we see that samples vary wildly independent of what we would expect from the omics. And that maybe is more aligned with what you see. So, the drugs do respond similarly across these samples, right? So like two EGFR inhibitors might not work in an EGFR mutant sample, right? But that's not to say that like they're not targeting EGFR in the same way. It's just saying that like this sample, for whatever reason, is not responding to EGFR inhibitors.
And maybe I will insert myself, Wes, with a quick question here, because I mean, you touched on a point that I think it's critical, which is most of these compounds are quote unquote dirty, right? I mean, they don't have a single target. Are you like purposely going after primary target? Do you have a model that incorporates secondary, tertiary, and other types of targets in there?
Yeah, so I didn't get into it how we actually chose target annotation, but yeah, so we use SelectChem. We actually found SelectChem to have like the best set of curated targets. We do some manual curation there. So Jess is actually a biophysicist and very deep in the drug world. So she did a lot of work on figuring out like, are these the right annotations for the targets? So yeah, drugs have multiple targets. The contrastive learning penalty considers every possible target that the drug might have. So if a drug is very dirty, it tries to move it towards all of the other drugs that also target some of those targets. But again, the data overwhelms it often.
Yeah, fair enough. Anyone from the chat that wants to speak up? Otherwise,
Maybe one quick question. Thank you very much for this talk. Hi, Alexander from Vienna, from the Medical University of Vienna. We work mostly on primary patient samples and do large high throughput drug screenings. You were showing the biological differences between cell lines and patient samples. Did you also look into organoids?
Yeah, absolutely. So I mean, so our data set contains a mixture of organoids and patient-derived cells. It's basically anything that's not a cell line, right? So for instance, like the pancreas samples are almost all organoids. It's like Dave Tuvison's and a few other groups organoid papers. So yeah, so depending on the cancer type, sometimes it's easier to build an organoid, as you guys know, right? Like sometimes it's easier to build an organoid, sometimes it's easier to just do patient-derived cells. Sometimes it's easier to do spheroids. Sometimes it's easier to do PDXs that you then harvest cells from. So depending on the cancer type, it might be a different ex vivo model that they have, but some of them are definitely organoids. Yeah.
And did you compare patient-derived direct ex vivo models and organoids about
Yeah, that's
clustering?
actually really difficult to do. I would love to do that. It's just there's not a ton of cancer types for which that comparison is available to us. Like it really does seem like there's, there's sort of like in pancreas, everyone does organoids, or or in yeah, I'm trying to think off the top of my head, but I can't. But yeah, there are other diseases where other cancer subtypes where like everyone just does short-term culture on their cells. Like there's just, and that makes sense, right? It's really hard to do organoids for certain cancer types.
Great, thank you.
Yeah.
All right. There is a question that I think is really interesting in the chat that points at tumor purity. I don't know who it is, so I'm happy to read it out. It says, does your model take into account tumor purity? We work with some samples where it's difficult to isolate truly pure malignant populations. And so how does your model account for that? And obviously, if you have long-term established organoids, you have some samples that are just tumor. And when you have more short-term, more primary samples, you have samples that are incredibly unpure, not pure. So do you account for that, Wes?
So yeah, that's a great question. This is where we kind of had to just take what's available, right? Like our model does not take that into account. For the most part, like it's not really published in most of these papers, like the tumor purity level of every single sample, for instance. Like that's just not data that we have available. What I would say is that, yeah, because it's this sort of mixture of lots of different kinds of ex vivo models, and what we see is sort of systematically across different models and cancer types, that sort of gives us the robustness hope that we're hoping that this is like a real effect. I would love to control for tumor purity, like we tried to control for additives and show that in fact, like this is a robust finding across tumor purity. But yeah, it's definitely a point in our discussion of our paper that we say, tumor purity is something we have to consider.
And I will say there was also a comment earlier, I don't remember, about the additives, Dennis, I think, about making sure that that list of additives you have, it's separated between factors that people use for generating organoids or models versus those that are used in drug screening. I will say from my view, you had a couple of things in there that we use when we process samples, but not growing them. So I think it may be worth going through that list. And I know it's a challenging work, and I assume many people are actually using proprietary media at this point, for which we don't have the exact composition. So I'm not saying you have to do that job, but it's a complex analysis, I guess.
Yeah, it is complex. I mean, and what we did was the union, right? So like a lot of things are kind of very vague in the methods descriptions, right? It's like very difficult to be very precise on a lot of this, I think, unless you're anticipating someone's going to do this. So yeah, so what Jess did was effectively do sort of like the union of anything that could possibly have been an additive used during the drug screen. And so in some sense, like it's true, we could distinguish between those two, but we tried controlling for each one of them, and none of them seems to really eliminate the finding.
Any other question?
Have you ever tried to do a CRISPR-Cas9 screen on the very same cell lines you did the drug screen and then correlate?
I literally yesterday had a call with our drug core to set up this experiment. So, and some of whom are on this call right now. So yeah, I'm extremely interested to do that.
Hi, Wesley, could I just jump in on that point there? Yeah, I was just kind of curious as well, using like the genomic, CRISPR screen plus drug. I was wondering it would be really useful to use this to reduce the space of a genetic screen. I'm just wondering what information you had to feed into Batchy. Was it the drug target, or was it's just the response? Because that could be great for like a CRISPR, what could we put in there to reduce the space that we'd need to screen?
Absolutely. So what's great about Batchy is it's model agnostic, works with anything, right? So it just looks at, you have a model, it has this uncertainty, I can quantify that and reduce that. So we can build the model in whatever way makes sense. The one we did in the paper is actually intentionally a very dumb model. It's basically a Bayesian factor model. It doesn't know anything about the drugs. It doesn't know anything about the samples. It just learns from the data. But yeah, it would make a lot of sense to include a lot of the existing biological knowledge we have for a CRISPR screen thing. I talked about this with some of the people that work on like Cas12 a long time ago to do combination CRISPR screens. And it is a little bit trickier because you can't really mix and match things quite as easily, as my understanding, in the CRISPR world as opposed to the drug world. So, but yeah, I'm super happy to explore that. Also, we don't have a wet lab in my lab, so if other people are like, "Yeah, you should explore that," like, and you have a wet lab, please talk to me.
Definitely will do.
Yeah.
Also, Wes, you can come volunteer in anyone's power lab anytime you want to learn how to pipette. No problem whatsoever. Listen, we are at the hour. Really appreciate the discussion, the thoughtful discussion, and the fantastic seminar by Wes. Thank you so, so much. And I think at this point we'll call it a day. Thank you again, and please remember, save the date. There is going to be an email going out. We're going to have a nice SFPM reception at AACR, and we'll see each other next month. Thank you so much.
Thank you, everyone.